Chapter 5, Principles of Inheritance and Variation. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animal, or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents, or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other, or sometimes even so different? These are these and several related questions are dealt with scientifically in branch of biology known as genetics. This subject deals with the inheritance as well as the variation of characters from parents to offspring. Inheritance is the process by which characters are passed on from parent to progeny. It is the basis of heredity. Variation is the degree by which progeny differs from their parents. Humans knew from as early as 8000 to 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variation that were naturally present in the wild population of plants and animals to selectively breed and select for organisms that possess desirable characters. For example, through artificial selection and domestication from ancestral wild cows, we have well known Indian breeds, for example, Sahipal cows in Punjab. We must, however, recognize that Pun- we must, however, recognize that though our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of these phenomena. 5.1 Mendel's law of inheritance. It was during the mid 19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on garden peas for 7 years from 1856 to 1863 and proposed the laws of inheritance in living organisms. During Mendel's investigations into inheritance patterns, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiments had a large sampling size which gave greater credibility to the data that he collected. Also, the confirmation of his inferences from experiments on successive generations of his test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposite traits, for example, tall or dwarf plants, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by the later scientists to account for all the diverse natural observations and the complexity inherent in them. Mendel conducted such artificial pollination or cross pollination experiments using several true breeding pea lines. A true breeding line is one that, having undergone continuous self continuous self-pollination, shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated full or constricted green or yellow pods, and tall or dwarf plants. Figure 5.1 7 pairs of contrasting traits in pea plants studied by Mendel. Character, dominant trait, recessive trait. Seed shape, round, wrinkled. Seed color, yellow, green. Flower color, violet, white. Pod shape, full, constricted. Pod color, green, yellow. Flower position, axial, terminal. Stem height, tall, dwarf. Table 5.1 Contrasting traits studied by Mendel in P. Characters constructing traits. Stem height, tall or dwarf. Flower color, violet or white. Flower position, axial or terminal. Pod shape, inflated or constricted. Pod color, green or yellow. Seed shape, round or wrinkled. Seed color, yellow or green. 5.2 Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel where he crossed tall and dwarf pea plants to study the inheritance of one gene. Figure 5.2 Steps in making a cross in pea. Petal, stigma, anther, stamen, carpel. removal of anthers, emasculation, parent female, parent male, transfer of pollination. He collected the seeds produced as a result of this process and grew them to generate plants of the first hybrid generation. This generation is called the first filial generation, first filial progeny, or F1. Mendel observed that all the F1 progeny plants were tall like one of its parent, none were dwarf. Figure 5.3. Diagrammatic representation of monohybrid cross. Parental tall cross dwarf plant. F1 generation tall cross tall selfing it. In F2 generation gives tall, tall, tall and a dwarf plant. He made similar observations for the other pairs of traits. He found out that F1 always resembled either one of the parents and that the trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants and to his surprise found that in the second filial generation some of the offspring were dwarf. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf were one-fourth of the F2 plants, while three-fourths of the F2 plants were tall. The tall and dwarf plants were the tall and dwarf traits were identical to their parental type and did not show any blending. That is all the offspring were either tall or dwarf. None were of in-between height. Similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in the F1 generation, while at the F2 generation both the traits were expressed in the proportion of 3:1. The contrasting traits did not show any blending at either F1 or F2 generation. F1 or F2 stage. Based on these observations, Mendel proposed that something was being stably passed down unchanged from parent to offspring through the gametes over the successive generations. He called he called these things as factors. Now we call them as genes. Genes, therefore, are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles, that is, they are slightly different forms of the same gene. If we use alphabetical symbols for each gene, that a capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character of height, T is used for the tall trait and small t for the dwarf, and capital T and small t are alleles of each other. Hence, in plants, the pair of alleles for height would be capital T, capital T, capital T, small t, or small t, small t. Mendel also proposed that in the true breeding tall or dwarf P variety, the allelic pair of genes for height are identical to homozygous capital T, and small t, respectively. Capital T, and small t are called the genotype of the plant, while the descriptive terms tall and dwarf are the phenotype. What then would be the phenotype of a plant that had a genotype capital T, small t? As Mendel found the phenotype of the F1 heterozygote T, capital T, small t, to be exactly like the capital T parent in appearance, he proposed that in a pair of dissimilar factors, one dominates the other, as in F1, and hence is called the dominant factor, while the other factor is recessive. In this case, capital T for tallness is dominant over small t for dwarfness, that is recessive. He observed identical behavior for all the other characters or trait pairs that he studied. It is convenient and logical to use the capital and lowercase of an alphabetical symbol to remember this concept of dominance and recessiveness. Do not use capital T for tall and small d for dwarf because you will find it difficult to remember whether capital D and small d are alleles of the same gene or character or not. Alleles can be similar as in the case of homozygous capital T and small t, or can be dissimilar as in the case of heterozygote capital T, small t. Since the capital T, small t plant is heterozygous for the genes controlling one character height, it is a monohybrid, and the cross between capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and the dwarf plant produces gametes by the process of meiosis, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to a gamete. This segregation of alleles is a random process, and so there is 50% chance of a gamete containing either allele, as has been verified by the results of the crossings in In this way, the gametes of the tall capital T plants have the allele capital T, and the gametes of the dwarf small t plants have the allele small t. During fertilization, the two alleles capital T from one parent, say through the pollen, and small t from the other parent, then through the egg, are united to produce zygotes that have one capital T allele and one small t allele. In other words, the hybrids have capital T small t. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents is the formation of the zygotes. The F1 and F2 plants can be understood from a diagram called a Punnett square as shown in figure 5.4. Figure 5.4, a Punnett square used to understand a typical monohybrid cross conducted by Mendel between two breeding tall plants and two breeding dwarf plants. Tall capital T cross dwarf small t gametes capital T small t F1 generation capital T small t 4 times selfing it, that is tall capital T small t gametes capital T small t F2 generation, which is capital T small t and small t. Phenotypic ratio tall is to dwarf 3 is to 1. Genotypic ratio, that is capital T is to capital T small t is to small t 1 is to 2 is to 1. It was developed by British geneticist Reginald C. Punnett and is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written on two sides, usually the top row and left columns. All possible combinations are represented in boxes below in the squares, which generates a square output form. A Punnett square shows the parental tall capital T male and dwarf small t female plants. The gametes produced by them and the F1 capital T small t progeny. The F1 plants of genotype capital T small t are self-pollinated. The symbols female and male are used to denote the female eggs and male pollen of the F1 generation respectively. The F1 plant of the genotype capital T small t, when self-pollinated, produces gametes of the genotype capital T and small t in equal proportion. When fertilization takes place, the pollen grains of the genotype capital T have a 50% chance to pollinate eggs of the genotype capital T as well as of the genotype small t. Also, pollen grains of the genotype small t have a 50% chance of pollinating eggs of genotype capital T as well as of genotype small t. As a result of random fertilization, the resultant zygotes can be of genotypes capital T, capital T, small t, or small t. From the Punnett square, it is easily seen that 1/4 of the random fertilization lead to capital T, half lead to capital T, small t, and 1/4 to small t. Though the F1 generation have a genotype of capital T, small t, but the phenotype character seen is tall at F2. 3/4 of the plants are tall, where some of them are capital T, while others are capital T, small t. As still it is not possible to distinguish between the plants with the genotypes capital T and capital T small t. Hence, within the genotypic pair capital T small t, only one character capital T tall is expressed. Hence, the character capital T or tall is said to dominate over the other allele small t or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall, though the genotype is capital T small t, and in the F2, 3/4 of the plants are tall, though genotypically 1/2 are capital T small t and only 1/4 are capital T. This leads to a phenotypic ratio of 3/4 tall, that is 1/4 capital T plus 1/2 capital T small t and 1/4 small t, that is 3:1 ratio, but a genotypic ratio of 1:2:1. The 1/4 is to 1/2 is to 1/4 ratio of capital T is to capital T small t is mathematically condensable to the form of binomial expression ax plus by the whole square, that has the gametes bearing genes capital T or small t in equal frequency of half. The expression is expanded as given below. Half capital T plus half small t the whole square is equal to half capital T plus half small t times half capital T plus half small t, which is equal to 1/4 capital T plus half capital T small t plus 1/4 small t. Mendel self-pollinated with the F2 plants and found that dwarf F2 plants contributed to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the dwarf was homozygous. Small t. What do you think he would have got had he self-pollinated a tall F2 plant? From the preceding paragraphs, it is clear that though the genotypic ratios can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 has capital T or small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed a tall plant from F2 with a dwarf plant. This he called a test cross. In a typical test cross, an organism P plants here showing a dominant phenotype and whose genotype is to be determined is crossed with the recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of test organism. Figure 5.5 shows the results of typical test cross where violet color of flower W is dominant over the white color of flower small w. Using Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get? Using the genotypes of this cross, can you give a general definition of a test cross? Figure 5.5 diagrammatic representation of a test cross. Homozygous recessive small w, homozygous recessive small w. Capital W times 4. Capital W, dominant phenotype, genotypic unknown. Capital W, small w, capital W, small w times 2, and small w times 2, which gives the allele of capital W and small w. Result in first case is all flowers are violet. Interpretation unknown flower is homozygous dominant. Result in second case, half of the flowers are violet and half of the flowers are white. Interpretation unknown flower is heterozygous. Based on his observation on monohybrid process, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid process. Today, these rules are called principles or laws of inheritance. The first law or law of dominance and the second law or law of segregation. 5.2.1 Law of dominance. First, characters are controlled by discrete units called factors. Second, factors occur in pairs. Third, in a dissimilar pair of factors, one member of the pair dominant, the other recessive. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. 5.2.2 Law of segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation. Though one of these is not seen at the F1 stage, though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. 5.2.2.1 Incomplete dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower snapdragon or Antirrhinium sp species is a good example to understand incomplete dominance. In a cross between true breeding red flowered R, and true breeding white flowered plants small r, the F1 capital R small r was pink. When the F1 was self-pollinated, the F2 resulted in the following ratio: 1 capital R red is to 2 capital R small r pink is to 1 small r white. That the genotype ratios were exactly as we would expect in any Mendelian monohybrid cross, but the phenotype ratios had changed from the 3:1 dominant or recessive dominant is to recessive ratio. What happened was that capital R was not completely dominant over small r, and this made it possible to distinguish capital R small r as pink from capital R red and small r white. Figure 5.6 Result of monohybrid cross in the plant of snapdragon where one allele is incompletely dominant over the other allele. Parental generation: red, capital R white, small r gametes capital R, small r. F1 generation, all pink, capital R, small r. Gametes, capital R, small r, capital R, small r. Female gamete, male gamete. F2 generation, capital R, capital R, capital R, small r, capital R, small r, small r, small r. Phenotypic ratio, red is to pink is to white, 1 is to 2 is to 1. Genotype ratio, capital R, capital R, capital R, small r, small r, small r, 1 is to 2 is to 1. Explanation of the concepts of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a developed organism, there are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not always be identical as in heterozygote. One of them may be different due to some changes that it has undergone, about which you will read further on and in the next chapter, which modifies the information that a particular allele contains. Let's take an example of a gene that contains the information for producing an enzyme. Now there are two copies of this gene, the two allelic forms. Let us examine as in more common that the normal allele produces the normal enzyme that is needed for the transformation of the substrate S. Theoretically, the modified allele could be responsible for the production of first, the normal or less efficient enzyme, or second, a non-functional enzyme, or third, no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele, that is, it will produce the same phenotype or trait, that is, the result in the transformation of substrate S. Such equivalent allele pairs are very common, but if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype or trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represents the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to the non-functional enzyme or because no enzyme is produced. 5.2.2.2 Codominance. Till now, we were discussing crosses where the F1 resembled either of the two parents' dominance or was in between incomplete dominance. But in the case of codominance, the F1 generation resembles both parents. A good example is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene I. Plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene, capital I, has three alleles, IA, IB, and small I. The alleles IA and IB produce a slightly different form of the sugar, while allele small I does not produce any sugar. Because humans are developed organisms, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over small I. In other words, when IA and small I are present, only IA expresses because small I does not produce any sugar. And when IB and small I are present, IB expresses, but when IA and IB are present together, they both express their own types of sugars. This is because of codominance. Hence, red blood cells have both A and B types of sugars. Since there are three different alleles, there are 6 different combinations of these three alleles that are possible, and therefore a total of 6 different genotypes of the human ABO blood types. Table 5.2. How many phenotypes are possible? Table 5.2 table showing the genetic basis of blood groups in human population. Allele from parent 1, allele from parent 2, genotype of offspring, blood type of offspring. IA, genotype of offspring, IA, blood type A. IB, AB. IA, small I, capital A. IB, IB, AB, blood group. IB, B, blood group. IB, small I, IBI, blood group B. Small I, blood group O. You realize that the example of ABO blood group also provides a good example of multiple alleles. Here you can see that there are more than 2, that is 3 alleles governing the same character. Since in an individual only 2 alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has 2 alleles, B and small b. Starch is synthesized effectively by capital B homozygotes, and therefore large starch grains are produced. In contrast, small b homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of the seeds, capital B seeds are round and the small b seeds are wrinkled. Heterozygotes produce the round seeds, and so capital B seems to be dominant allele. The starch grains produced are of intermediate size in capital B small b seeds. So if starch grain size is considered as the phenotype, and from this angle the alleles is showing complete dominance. Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of the particular phenotype from this product as it does not particular phenotype that we choose to examine, as it does on the particular phenotype that we choose to examine. In case more than one phenotype is influenced by the same gene. 5.3 Inheritance of two genes. Mendel also worked with and crossed pea plants that differed in two characters as is seen in the cross between a pea plant that has the seeds with yellow color and round shaped and one that had seeds of green color and wrinkled shape. Mendel found that the seeds resulting from the crossing of the parents had yellow color and round shaped seeds. Here you can tell which of the characters in the pair's yellow or green color and round or wrinkled shape was dominant. Thus, yellow color was dominant over green and round shape dominant over wrinkled. These results were identical to those and that he got when he made a separate monohybrid process between yellow and green seeded plants and between round and wrinkled seeded plants. Let us examine the genotype symbols. Capital Y for dominant yellow seed color and small y for recessive green seed color. Capital R for round shaped seeds and small r for wrinkled seed shape. The genotype of the parents can then be written as capital R, capital Y, and small r, small y, small y. The cross between the two plants can be written down as in figure 5.7, showing the genotypes of the parent plants. The gametes capital R, capital Y, and small y unite on fertilization to produce the F1 hybrid capital R, capital Y, small y. When Mendel self-hybridized the F1 plants, he found that 3/4 of the F2 plants had yellow seeds and 1/4 had green. The yellow and green color segregated in a 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. Figure 5.7 Result of a dihybrid cross between where the two parents differed in two pairs of contrasting traits, seed color and seed shape. Parent generation: round yellow, capital R, capital Y, wrinkled cross, wrinkled green, small r, small y, gametes, capital R, capital Y, cross, small r, small y, round yellow, capital R, capital Y, small y, F1 generation, selfing it, gametes, small r, capital Y, small r, capital Y, capital R, small y, small y, of female and same with males. F2 generation gives capital R, capital Y, capital Y, capital R, capital small r, capital Y, 2 times. Capital R, capital Y, 2 times. Capital R, capital Y, small y, 4 times. Small r, capital Y, small y, 2 times. Capital R, small y, 2 times. Small r, small y, 1 time. Capital R, small y, 1 time. Small r, capital Y, 1 time. Phenotypic ratio: round yellow 9, round green is to round green 3 is to wrinkled yellow 3 is to wrinkled 1, wrinkled green 1. Law of independent assortment. In the dihybrid cross, the phenotypes round yellow, wrinkled yellow, round green and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination of series of 3 yellow, 1 green with 3 brown is to 1 wrinkled. This observation can be written as follows: 3 brown is to 1 wrinkled, 3 yellow is to 1 green is equal to 9 brown yellow is to 3 wrinkled yellow is to 3 round green is to 1 wrinkled green. Based upon such observation on dihybrid crosses between plants differing in 2 traits, Mendel proposed a second set of generalization that we call Mendel's law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. The Punnett square can be effectively used to understand the independent segregation of the two pairs of the genes during meiosis and the production of eggs and pollen in the F1 capital R small y capital R small r capital Y small y plant. Considered the segregation of one pair of genes capital R and small r. 50% of the gametes have the gene capital R and the other 50% have small r. Now, besides each gamete having either capital R or small r, it should also have allele capital Y or small y. The important thing to remember here is the segregation 50% capital R and 50% small r is independent from the segregation 50% capital Y and 50% small y. Therefore, 50% of small r bearing gametes has capital Y and other 50% has small y. Similarly, 50% of the capital R bearing gametes has capital Y and other 50% has small y. Thus, there are four types of genotypes of gametes, four types of pollen, and four types of eggs. The four types are capital R, capital Y, small r, capital Y, and small y, each with a frequency of 25% or one-fourth of the total gametes produced. When you write down the four types of eggs and pollen of the two sides of a Punnett square, it is very easy to derive the composition of the zygote that gives rise to F2 plants. Although there are 16 squares, how many different types of genotypes and phenotypes are formed? Note them down in the format given. Can you, using the Punnett square data, work out the genotypic ratio at the F2 stage and fill in the format given in? Is the genotypic ratio also 9:3:1? 5.3.2 Chromosomal theory of inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons it remained unrecognized till 1900s. Firstly, communication was not easy as it is now in those days, and his work could not widely be publicized. Secondly, his concept of genes or factors in Mendel's words as stable and discrete units that control the expression of traits and of the pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factors genes were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. In 1900s, three scientists, De Vries, Correns, and von Schirmack, independently rediscovered Mendel's results on inheritance of characters. Also, by this time, due to the advancement in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appear to double and divide just before each cell division. These were called chromosome-colored bodies as they were visualized by staining. By 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of chromosome was parallel to the behavior of genes and used chromosome movement to explain Mendel's law. Recall that you have studied the behavior of chromosome during mitosis, equational division, and during meiosis, reductional division. The important things to remember are that chromosome as well as genes occur in pairs. The two alleles of a gene pair are located on homologous sites on homologous chromosome. Figure 5.8 Meiosis and germ cell formation in a cell with 4 chromosomes. Can you see how chromosomes segregate when germ cells are formed? G1, G2, Meiosis 1 anaphase, bivalent. Meiosis 2 anaphase, germ cell. Figure 5.3 A comparison between behavior of chromosomes and gene. Chromosome occur in pair, gene occur in pair. Chromosome segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete. Gene segregate at gamete formation and only one of each pair is transmitted to a gamete. Chromosome independent pairs segregate independently of each other. One pair segregate independently of another pair in gene. Can you tell which of these column A or B represent the chromosome and which represent the gene? How did you decide? During anaphase of meiosis 1, the two chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosome of four different colors in the left and right columns. In the left column, possibility 1 orange and green is segregating together, but in the right hand column, possibility 2 the orange chromosome is segregating with the red chromosome. Possibility 1 one long orange and short green chromosome and long yellow and short red chromosome at the same pole. Possibility 2 one long orange and short red chromosome and long yellow and short green chromosome at the same pole. Meiosis 1 of anaphase, meiosis 1 of anaphase. Meiosis 2 anaphase, meiosis 2 anaphase. Germ cells. Sutton and Boveri argued that the pairing and separation of the pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Following the synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to the discovering the basis for the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies Drosophila melanogaster, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They completed their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes. The male and the female flies are easily distinguishable. Also, it has many types of hereditary variations that can be seen with low power microscopes. Figure 5.10 Drosophila melanogaster A male, B female. 5.3.3 Linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex linked. The crosses were similar to the dihybrid crosses carried out by Mendel in bees. For example, Morgan hybridized yellow-bodied, white-eyed females to brown-bodied, red-eyed males and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and F2 ratio deviated very significantly from 9:3:3:1 ratio expected when the two genes are independent. Morgan and his group knew that the genes were located on the X chromosome and saw quickly that when the two genes in dihybrid crosses were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on the chromosome, and the term recombination to describe the generation of non-parental gene combinations. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, showed very low combination, figure 11, figure 5.11 cross A, while others were loosely linked, showed higher combination, figure 5.11 cross B. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2% recombination. His student Alfred Sturtevant used a frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genomes, as was done in the case of human genome sequencing project described later. Figure 5.11 linkage. Results of two dihybrid crosses conducted by Morgan. Cross A shows a crossing between gene Y and gene small y and capital and small w. Cross B shows a crossing between genes small w and small m. Here dominant wild type alleles are represented with plus sign in superscript. Note the strength of linkage between small y and small w is higher than small w and small m. Cross A female male parental yellow white male wild type F1 generation female and male gametes F2 generation parental type 98.7%, recombinant type 1.3%. Wild type white, yellow, white, yellow, wild type, white, yellow. Cross B, female, male, white, miniature, wild type, parental, F1 generation, wild type, white, miniature, gametes, parental type 62.8%, recombinant type 37.2%, wild type, miniature, white, wild type, miniature, white. 5.4 Polygenic inheritance. Mendel's studies mainly describe those traits that have distinct alternate forms, such as flower color, which are either purple or white. But if you look around, you will find out that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we don't just have tall or short people as two distinct alternatives, but a whole range of possible heights. Such traits are generally controlled by three or more genes and are thus called as polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele, that is, the effect of each allele is additive. To understand this better, let us assume that three genes A, B, C control skin color in human, with the dominant forms A, B, and C responsible for dark skin and the recessive forms a, small b, and small c for light skin color. The genotype with all the dominant alleles A, capital B, capital C will have the darkest skin color, and that of with all the recessive alleles a, small b, small c will have the lightest skin color. As expected, the genotype with three dominant alleles and three recessive alleles will have an intermediate skin color. In this manner, the number of each type of alleles in the genotype would determine the darkness or lightness of the skin in an individual. 5.5 Pleiotropy. We have so far seen the effect of a gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotypic expressions. Such a gene is called pleiotropic gene. The underlying mechanism of pleiotropy in most cases is the effect of a gene on metabolic pathways which contribute towards different phenotypes. An example of this is the disease phenylketonuria, which occurs in humans. The disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase. Single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation and a reduction in hair and skin pigmentation. 5.6 Sex determination. The mechanism of sex determination has always been a puzzle before the geneticist. The initial clue about the genetic or a chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observation made in a number of insects led to the development of the concept of genetic or chromosomal basis of sex determination. Henking in 1891 could trace the specific nuclear structure of of through all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Henking gave a name to this structure as X body, but he could not explain its significance. Further investigation by other scientists led to the conclusion that the X body of Henking was in fact a chromosome, that and that is why it was given the name X chromosome. It was also observed that in a large number of insects, the mechanism of sex determination is of the XO type, that is, all eggs bear an individual X chromosome besides the other chromosome autosome. On the other hand, some of the sperms bear the X chromosome, whereas some do not. Eggs fertilized by sperm having an X chromosome become female, and those fertilized by sperms that do not have an X chromosome becomes male. Do you think the number of chromosomes in the male and female are equal due to the involvement of the X chromosome in the determination of sex? It was designated to be the sex chromosome, and the rest of the chromosomes were named as the autosome. Grasshopper is an example of XO type of sex determination in which the males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosomes. These observations led to the investigation of a number of species to understand the mechanism of sex determination in a number of other insects and mammals, including man. XY type of sex determination is seen where both male and female have same number of chromosome among the males, and X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosome, but both male and female bear the same number of autosomes, hence the male have autosomes plus XY, while females have autosomes plus XX. In human beings and in Drosophila, the males have one X and one Y chromosome, whereas females have a pair of X chromosomes besides autosome. In the above description, you have studied about two types of sex determining mechanism, that is XO type and XY type. But in both cases, male produce two different types of gametes, A, either with or without X chromosome, or B, some gametes with X chromosome and some with Y chromosome. Such type of sex determination mechanism is designated to be the example of male heterogamety. In some other organisms, for example birds, a different mechanism of sex determination is observed. In this case, the total number of chromosomes is same in both males and females, but two different types of gametes in terms of the sex chromosome are produced by females, that is female heterogamety. In order to have a distinction with the mechanism of sex determination described earlier, the two distinct sex chromosome of a female bird has been designated to be Z and W chromosome. In these organisms, the female one have Z and one W chromosome, whereas male have a pair of Z chromosomes besides the autosomes. Figure 5.12, determination of sex by chromosome differences. A, B, both in humans and in Drosophila, the female has a pair of XX chromosome homogametic and the female XY heterogametic composition. C, in many birds, female has a pair of dissimilar chromosome ZW and male two similar ZZ chromosomes. 5.6.1, sex determination in humans. It has already been mentioned that the sex determining mechanism in the case of humans is XY type. Out of 23 pairs of chromosomes present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosomes are present in the female, whereas the presence of an X and Y chromosome are determinant of the male characteristic. During spermatogenesis among males, two types of gametes are produced. 50% of the total sperm produced carry gametes are two types of gametes. 50% of the total sperm produced carry the X chromosome, and the rest of 50% has Y chromosome besides the autosomes. Female, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. In case the ovum fertilizes with a sperm carrying X chromosome, the zygote develops into a female XX, and the fertilization of ovum with Y chromosome carrying sperm results into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or a female child. It is unfortunate that in our society women are blamed for giving birth to female children and have been ostracized and ill-treated because of this false notion. 5.6.2, sex determination in honeybee. The sex determination in honeybee is based on the number of sets of the chromosome an individual receives. An offspring formed from the union of a sperm and an egg develops as a female queen or worker, and an unfertilized egg develops as a male drone by means of parthenogenesis. This means that the males have half the number of chromosomes that of a female. The females are diploid having 32 chromosomes, and males are haploid, that is having 16 chromosomes. This is called as haplodiploid sex determination system and has special characteristic features such as the males produce sperms by mitosis. They do not have father and thus cannot have sons, but have grandfather and can have grandsons. How is the sex determination mechanism different in the birds? Is the sperm or the egg responsible for the sex of the chicks? Figure 5.13, sex determination in honeybee. Parents female 32, male 16, meiosis, baby 16, F1 generation male 16. Parents male 16, mitosis, gametes 16, F1 generation female 32. Mutation. 5.7, mutation. Mutation is a phenomenon which results in alternation of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variation in DNA. As we will learn in chapter 6, one DNA helix runs continuously from one end to the other in each chromatid in a highly supercoiled form. Therefore, loss, deletions, or gain insertion or duplication of a segment of DNA result in alternation in chromosome. Since genes are known to be located on chromosome, alternation in chromosome result in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. In addition to the above, mutation also arise due to change in a single base pair of DNA. This is known as point mutation. A classical example of such a mutation is a sickle cell anemia. Deletion and insertion of base pairs of DNA causes frame shift mutation. See chapter 6. The mechanism of mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induces mutations. These are referred to as mutagens. UV radiations can cause mutations in organisms. It is a mutagen. Figure 5.13. Symbols used in human pedigree analysis: male, female, sex unspecified, affected individuals, mating, mating between relatives, consanguineous mating, parents above and children below in order of birth left to right, parents with male child affected with disease, 5 unaffected offspring. 5.8 Genetic disorders. 5.8.1 Pedigree analysis. The idea that disorders are inherited and has been prevalent in the human society since long. This was based on the heritability of the certain characteristic features in females. After the rediscovery of Mendel's work, the practice of analyzing inheritance pattern of traits in human beings began, since it is evident that control process that can be performed in pea plants or some other organisms are not possible in case of human beings. Study of the family history about human beings. Study of the family history about inheritance of a particular trait provides an alternative. Such an analysis of traits in the several generations of a family is called the pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over a generation. In human genetics, pedigree studies provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality, or a disease. Some of the important standard symbols used in the pedigree analysis have been shown in Figure 5.13. As you have studied in this chapter, each and every feature in an in any organism is controlled by one or the other gene located on the DNA present in the chromosome. DNA is the carrier of genetic information. It is transmitted from one generation to the other without any change or alteration. However, changes or alteration do not take place occasionally. Such an alteration or change in the genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with the inheritance of change or altered genes or chromosome. 5.8.2 Mendelian disorders. Broadly, genetic disorders may be grouped into two categories: Mendelian disorders and chromosomal disorders. Mendelian disorders are mainly determined by alternation or mutation in the single gene. These disorders are transmitted to the offspring on the same lines as we have studied in the principle of inheritance. The pattern of inheritance of such Mendelian disorders can be traced in a family of the pedigree by the pedigree analysis. Most common and prevalent Mendelian disorders: hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome. As in case of hemophilia, it is evident that X-linked recessive trait shows transmission from carrier female to male progeny. A representative pedigree is shown in Figure 5.14 for dominant and recessive traits. Discuss with your teacher and design pedigrees for characters linked to both autosomes and sex chromosome. Figure 5.14 Representative pedigree analysis of a autosomal dominant trait, for example, myotonic dystrophy. B. Autosomal recessive trait, for example, sickle cell anemia. Color blindness, it is a sex-linked recessive disorder due to defect in either red or green cone of the eye, resulting in failure to discriminate between red and green color. This defect is due to mutation in certain genes present in the X chromosome. It occurs in about 8% of males and only about 0.4% of females. This is because the genes that led to red-green color blindness are on the X chromosome. Males have only one X chromosome and females have two. The son of a woman who carries the gene has 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. Hemophilia, this sex-linked recessive disease which shows in transmission, its transmission from unaffected carrier female to some of the male progeny has been widely studied. In this disease, a single protein that is a part of the cascade of proteins involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. Heterozygous Heterozygous female carrier for hemophilia may transmit the disease to sons. The possibility of a female becoming a hemophilic is extremely rare because mother of a such female has to be at least a carrier and the father should be hemophilic and viable in the later stages of life. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. Sickle cell anemia, this is an autosome linked recessive trait that can be transmitted from parents to the offspring when both the partners are carrier for the gene or heterozygous. The disease is controlled by a single pair of allele, HBA and HBS. Out of the three possible genotypes, only homozygous individuals for HBS show the diseased phenotype. Heterozygous HBA HBS individuals appear apparently unaffected, but they are carrier of the disease, as there is 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. The defect is caused by the substitution of glutamic acid GLU by valine BAL at the sixth position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to single base substitution at the sixth codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. Figure 5.15 micrograph of the red blood cells and the amino acid composition of the relevant portion of beta chain of hemoglobin. A from a normal individual, B from a individual with single cell anemia. Normal HBA gene, GAG, CTC, GAG mRNA, Val1, His2, Leu3, THR4, Pro5, Glu6, Glu7, HBA peptide, sickle cell HBS gene, GTG, CAC, mRNA, GUG, Val1, HIS2, Leu3, THR4, Pro5, Val6, GLU, glutamic acid 7, HBS peptide. Phenylketonuria, the inborn error of the metabolism, is also inherited as an autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenyl pyruvic acid and other derivatives. Accumulation of these in brain results in mental retardation. These are also excreted through urine because of its poor absorption by kidney. Thalassemia, this is also an autosome linked to recessive blood disease transmitted from parents to the offspring when both the partners are unaffected carrier for the gene or heterozygous. The defect could be due to either mutation or deletion, which ultimately results in the reduced rate of synthesis of one of the globin chains, alpha and beta chains that make up hemoglobin. This causes the formation of abnormal hemoglobin molecules, resulting into anemia, which is characteristic of the disease. Thalassemia can be classified according to which chain of the hemoglobin molecule is affected. In alpha thalassemia, production of alpha globin chain is affected, while in beta thalassemia, production of beta globin chain is affected. Alpha thalassemia is controlled by two closely linked genes HBA1 and HBA2 on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of the four genes. The more genes affected, the less alpha globin molecules produced, while beta thalassemia is controlled by a single gene HBB on chromosome 11 each parent and occurs due to mutation of one or both the genes. Thalassemia differs from sickle cell anemia in that the former is a quantitative problem of synthesizing too few globin molecules, while the later is a qualitative problem of synthesizing an incorrectly functioning globin. 5.8.3 Chromosomal disorders. The chromosomal disorders, on the other hand, are caused due to the absence or excess of or abnormal arrangement of one or more chromosomes. Failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosomes called aneuploidy. For example, Down syndrome results in the gain of extra copy of chromosome 21. Similar to Turner syndrome results due to loss of an X chromosome in human females. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. This condition is often seen in plants. The total number of chromosomes in a normal human cell is 46, 23 pairs. Out of these, 22 pairs are autosomes and one pair of chromosomes are sex chromosomes. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one or any one pair of chromosomes. These situations are known as trisomy or monosomy of a chromosome, respectively. Such a situation leads to a very serious consequence in the individual. Down syndrome, Turner's syndrome, Klinefelter's syndrome are common examples of the chromosomal disorders. Down syndrome. The cause of this genetic disorder is the presence of an additional copy of the chromosome number 21, trisomy of 21. This disorder was first described by Langdon Down, 1866. The affected individual is short-statured with small round head, furrowed tongue, and partially open mouth. Figure 5.16. Palm is broad with a characteristic palm crease. Physical, psychomotor, and mental development is retarded. Figure 5.16, a representative figure showing an individual inflicted with Down syndrome and a corresponding chromosome of the individual. Flat back of the head, broad, flat face, big and wrinkled tongue, congenital heart disease, many loops on fingertips, palm crease. Klinefelter syndrome. This genetic disorder is also caused due to the presence of an additional copy of X chromosome resulting into a karyotype of 47XXY. Such an individual has overall masculine development. However, the feminine development of breast, that is gynecomastia, is also expressed. Such individuals are sterile. Figure 5.17. Diagrammatic representation of genetic disorders due to sex chromosome composition in humans. A. Klinefelter syndrome. Tall stature with feminized character. Turner's syndrome. Such a disorder is caused due to the absence of one of the X chromosome, that is 45, with X not such X0. Such females are sterile as ovaries are rudimentary besides other features including lack of other secondary sexual characters. Figure 5.17b. Turner's syndrome. Short stature and underdeveloped feminine character. Summary. Genetics is a branch of biology which deals with the principle of inheritance and its practices. Progeny resembling their parents in a morphological and physiological features has attracted the attention of many biologists. Mendel was the first to study this phenomenon systematically. While studying the pattern of inheritance in pea plants of contrasting characters, Mendel proposed a principle of inheritance, which are today referred to as Mendel's law of inheritance. He proposed that the factors later named as genes regulating the characters are found in pairs known as alleles. He observed that the expression of the characters in the offspring follow a definite pattern in different first generation F1 and second F2 and so on. Some characters are dominant over others. The dominant characters are expressed when factors are in heterozygous condition. Law of dominance. The recessive characters are only expressed in homozygous conditions. The characters never expressed in heterozygous condition. A recessive character that was not expressed in heterozygous condition may be expressed again when it becomes homozygous. Hence, characters segregate while formation of the gametes. Law of segregation. Not all characters show true dominance. Some characters show incomplete and some show co-dominance. When Mendel studied the inheritance of two characters together, it was found that the factors independently sort and combine in all the permutation and combination of independent assortment. Different combination of gametes are theoretically represented in a square tabular form known as Punnett square. The factors now known as gene and chromosome regulating the characters are called the genotype, and the physical expression of the characters is called the phenotype. After knowing that the genes are located on the chromosome, a good correlation was drawn between the Mendel's law, segregation and assortment of chromosomes during meiosis. The Mendel's law was extended in the form of chromosomal theory of inheritance. Later, it was found that Mendel's law of independent assortment does not hold true for the genes that were located on the same chromosome. These genes were called as the linked genes. Closely located genes assorted together and distantly located genes due to a combination of assorted independently. Linkage maps therefore correspond to arrangement of genes on a chromosome. Many genes were linked to sexes also and called as sex-linked genes. The two sexes, male and female, were found to have a set of chromosomes which were common and another set which was different. The chromosome which were different in two sexes was named as sex chromosome. The remaining set was named as autosomes. In humans, a normal female has 22 pairs of autosomes and a pair of sex chromosomes XX. A male has 22 pairs of autosomes and a pair of sex chromosomes as XY. In chicken, sex chromosome in male are ZZ and in females are ZW. Mutation is defined as a change in the genetic material. A point mutation is a change of single base pair in DNA. Sickle cell anemia is caused due to change of one base in the gene coding for beta chain of hemoglobin. Inheritable mutations can be studied by generating a pedigree of a family. Some mutations involve changes in a whole set of chromosome polyploidy or change in a subset of chromosome number aneuploidy. This helps in understanding the mutational basis of genetic disorders. Down syndrome is due to trisomy of chromosome 21, where there is an extra copy of chromosome 21, and consequently the total number of the chromosome becomes 47. In Turner's syndrome, one X chromosome is missing and the sex chromosome is as XO. And in Klinefelter's syndrome, the condition is XXY. These can be easily studied by analysis of karyotypes.